Hello and welcome to Organ on a Chip mini series. And we're going to begin this with what is organ on a chip and why was it developed? Let's take a closer look at the drug development process. Now, it begins with drug discovery, where thousands of chemicals are explored, and then comes preclinical testing. Now, this is a stage where animal testing is still unjustifiably a regulatory requirement. This is supposedly the gatekeeper for what's safe and effective enough to move forward into humans. If it passes the stage, the drug then enters clinical trials where it's tested on humans in several phases. These trials check how well it works, how safe it is, and whether the benefits outweigh the risks. Now, finally, if all of these trials are successful, the company can then apply for regulatory approval. Now, of all the drugs that enter human clinical trials, meaning they've already passed animal testing, about 90% fail. Now, that's the 90% statistic you often hear. It reflects the failure rate from animal testing to human studies. But if we look at the process from animal testing to the market, i.e., to the pharmacy, to your medicine cabinet, only about 5% of drug candidates ever make it to that market, to your pharmacy. That's a 95% failure rate. So even after passing animal tests, 95% of drugs still fail once we try and bring them to the market. That's not a small a margin of error. This is a system wide failure. And if something takes 10 years and over $3 billion, we need to be looking at why are we still using animal tests. Now, non-animal alternatives are not just a choice, but a scientific necessity. One of the fundamental reasons why we can't use the outcomes of animal tests to relate to human outcomes is because animals are not tiny humans. Their biology reacts very different to medicines. And as a result, some drugs that look safe in animals can prove harmful or deadly or ineffective in people, while promising treatments sometimes get abandoned too soon. And that mismatch wastes time, money, and delays life saving therapies. And so scientists realize we need to find a way to model human biology effectively in the laboratory, a way that could speed up discovery, make it more cost effective, and also create new medicines that are effective and are relevant to human patients. At the Weiss Institute at the University of Harvard, a team of engineers, biologists, and clinicians joined forces to invent a new approach. They borrowed techniques used to make computer microchips and adapted them to build organ on a chip devices. One of the founding pioneers of this technology, Don Ingber, put it simply: we took a game changing advance in microengineering made in our academic lab, and in just a handful of years, they turned it into a technology that is now poised to have a major impact on society. Now, that was almost 2 decades ago, and it's since been commercialized and used in multiple types of organs all over the world in different disease areas. And the impact of an organ on a chip, quite simply, it does not involve animals. And it does not harm patients either, because you're looking at the organ outside of the body. Coming up next, we're going to meet the chip and look at its components and see how it creates the living organ. Thank you for listening.